Hi, I'm Nitya Ramakrishnan and I'm here to present our work on epigenetic inheritance and information theoretic model. Why do skin cells behave differently from neurons in spite of having the same genetic code, that is DNA? It is because on top of the genetic information, there are several layers of epigenetic information that define the functions of cells as well as maintain their state. These epigenetic phenomena are responsible for turning on or off certain genes and providing specific functionality to the cells. An important set of epigenetic information is the chemical modifications to the histones, the binder proteins that keep the DNA compact. Did you know that the DNA replicates more than 1 trillion times every day in humans? Every time a mother chromatin replicates into two daughter strands, the epigenetic modifications have to be inherited and maintained in both the daughter strands. How the modification patterns are correctly copied from the mother to the daughter strands in order to maintain the state of the cell is a puzzle. In our work, we attempt to answer this puzzle using parallel concepts from communication theory. In telecommunication, a signal from the transmitted end is subjected to noise in the channel in which it is sent to the receiver. At the receiving end, the noisy received signal is decoded to obtain a signal that is almost the same as a transmitted signal. Techniques from information encoding theory are employed to construct codes that help in the detection and correction of the signal in the receiver. Similarly, the enzyme machinery attempts to reconstruct the mother chromatin's pattern in the daughter chromatids using transgenerational memory. We propose a threshold K-filling algorithm simplified from communication principles for this reconstruction as follows. Once the daughter chromatin is formed, the parental histones with the modifications intact are randomly placed at various positions. The newly generated histones without the modifications are placed in between the parental histones. Our model proposes that enzyme machinery modifies the unmodified histones in the gaps between the parental histones to match them with your neighbors as long as the gaps are less than a threshold K in length. In this case, for example, we see that gaps of 1 or 2 are filled, whereas gaps of 6 are unfilled. This is an initial reconstruction technique which, with aided by secondary mechanisms like DNA methylation maintenance throughout the cell cycle, would attempt to reconstruct a mother-like pattern in the daughter sequence. The exact value of the threshold here depends on the statistical nature of the mother chromatin. In our work, using simulations and verification with experimental data, we show how this algorithm is successful in reconstructing the original pattern and thereby preserving the epigenetic state across several generations. Thank you.